My name's Christina Mitchell. I'm Dean of the Faculty of Medicine, Nursing and Health Sciences at Monash University. And this is the plenary session on translational research, advancing innovative treatment. So we are very blessed to have 5 wonderful international speakers today to give us their insights into translational research and how to translate early discoveries into new drugs, products and changes in clinical practice. Central to this commercialization pipeline and translation is a valley of death that both investigators and commercialization agents speak about. It's the gap between taking a great discovery into clinical practice. Today, our 5 international speakers have navigated or are navigating this dangerous passage and will tell their stories. So we are very lucky today. Our first speaker is Professor Elizabeth Blackburn, the President of the Salk Institute for Biological Studies in California. Professor Blackburn won the Nobel Prize for Medicine in 2009 for discovering the molecular nature of telomeres, structures that are essential for maintaining and regulating our DNA. In addition to the Nobel Prize, Professor Blackburn has received nearly every major biomedical science and medical research award, including the Lasker, the Gruber and Garnier Prizes. She was named in the Time 100 Most Influential People in 2007. She's a member of numerous prestigious scientific societies, and I could take the whole 25 minutes of her talk to tell you how fabulous she is, but it's probably better to get her started, and the title of her talk is "It Really Is a Two-Way Street." Thank you.
Thank you, Christina, and thank you so much for inviting me to be part of this really wonderful summit. Right away, to dispel any mystery, what I'm referring to in my title of my talk was something that I really feel very much is important to keep in mind as we think about translation, and that is that while we usually think, as Christina described, from bench to bedside, what I think is very important is from bedside, from the human, to come back to understanding what it is going on in the disease processes that are such, a major part of the challenge for world health, and then to be able to continually refine and understand what one learns from humans back into the laboratory and then back into clinical practice. And so I'm going to give a few examples of that. So let's put a very general perspective out, first of all. Clearly, worldwide, in all parts of the world, there is a general aging demographic, and we know that a large number of influences feed into what are becoming some of the most major diseases that become more and more prevalent as some of the challenges to health, such as infectious diseases, and by no means is that solved, but as some of those become, in hand better, we now see revealed more and more age-related diseases. And there's quite a variety. I've listed them here on the bottom of the slide as some strong examples of what afflicts humans as they get older, and it can cause mortality in certain cases of these examples. But what's interesting is the biology of, why sometimes they look so different. Cardiovascular will be affecting one part of the, certain organs. Diabetes will apparently be, primarily in different organs. But in fact, what's very interesting is they often go hand in hand. So what is the underlying biology of that? And that can be related to the question of how do we age, and the process of aging that is prominently, as we just saw, is a prominent part of aging is, of course, increased susceptibility to all sorts of diseases with time, and that is part and parcel of what is the underlying biology of aging, and how does that play out in humans? And so I point this general context out because our own research, which is into telomeres, the ends of chromosomes that protect the ends of the DNA and hence the genetic material on on every chromosome, at every end of every chromosome. That's the research that, from very basic science beginnings that I've been involved in and was involved in quite early in the molecular studies of these, that research then has given us some information about that aspect of aging that I referred to, the increased susceptibility to diseases with time. So I want to just relate the story a little bit, first of all, with regard to the bench to bedside, and then you'll see how this comes to play back as well into the other way around. So telomeres, very briefly, cap the ends of chromosomes. They consist of highly repeated DNA sequences that form a scaffold upon which a sheath of well-understood protective proteins form, and this cap of repeated DNA sequences, typically 10,000 base pairs, for those of you who like numbers, at the ends of our chromosomes when we're born, that makes this sheath for this whole set of proteins, which are protective, as I said, but which, if the tract of repeated sequences gets too short, this cap can't form properly if the ends erode away, which they do through a variety of now quite well understood processes, such as DNA end replication being incomplete from replication stress that occurs as replication forks move through telomeric repeats, oxidative damage, chemical and other damage. All these things can combine in various settings to make telomeres get shorter, and when that happens, they can't cap the ends of chromosomes properly. Now, in humans, what happens is that over the many decades of human life, in fact, this dwindling down does happen overall on average in many, if not most, cell types. And so the question was, what does that do at the cellular level? There's a great deal now that's understood about it, and I'm going to illustrate at one level, the cell level, an example of what happens. So what happens is that if we look at a human cell, this beautiful micrograph where the purple is the chromosomes and the green lights up the mitochondria, the energy-forming units in the energy factories in the body, and the telomeres you can't see, but of course they're at the ends of the chromosomes. What happens is that if for all these reasons telomeres get shorter, at some point the telomere will become non-functional. That's actually when it has about 3.8 kilobases of DNA repeated sequences worth at the ends. So it's still got some repeated sequences, but that sheath of proteins no longer protects. That uncapped state then sets up a whole program in the cell which involves transcriptional changes. Big changes go on. The cell is reprogrammed. It's called a senescent state sometimes. One of the things that can happen is that the control of mitochondria, because of these transcriptional and gene expression changes, the control of mitochondria changes, and long story short, increased reactive oxygen species. Now that sets up a vicious cycle because telomeric DNA, for reasons best known to nature, but it's curiously susceptible to reactive oxygen species compared with the rest of the genome, so that'll provoke even more shortening and loss. And the other kind of consequence is that the program that is set up in cells makes increases in pro-inflammatory, even pro-tumorigenic factors, which is secreted. So now these cells can have paracrine effects as well as autocrine effects. So this is just one example of the kinds of things that happen when telomeres are uncapped, and this is quite well understood at the cellular level. And that the increased pro-inflammatory environment again sets up increased reactive oxygen species. So you've got a kind of vicious cycle. The enzyme telomerase, which we discovered in the 1980s, has the enzymatic job of adding repeats to the ends so that you get elongation. And so there is this activity, it exists. Now, the mechanism of it we understand quite well. It's copying RNA in the enzyme ribonucleoprotein complex into DNA. But what has been learned from decades of work in human cells of all different kinds is that this activity is under extraordinarily complex and tight controls in human cells. And at every molecular level you can think of, there will be controls, usually downwards, of telomerase, but for obviously upregulation in certain situations. Now, what we know in humans then is that you need enough telomerase to get you through a normal, healthy life. So what you learn from genetics is, in its simplest form, if you have a mutation that simply reduces the level of telomerase by half, or reduces the ability of those protective proteins to work completely. Just reduces them, doesn't knock it out, but just reduces it. Very clearly, this causality that shortens telomeres. That has been shown in a lot of work very clearly to be causally what's causing impacts on certain diseases, and there's a range of these diseases which you will immediately recognize as having characteristics with those that overlap those that afflict us more and more as we age. Unfortunately, for the individuals with these diseases, these are very severe and often early onsets of them, and there are different combinations in different individuals. Clearly these effects are happening system-wide, but how they play out can vary a great deal in terms of clinical presentations, but the causality is really clear. And just for those of you who like a bit more specificity, there's now 11 genes very well documented in which a mutation in these genes dedicated to telomerase or dedicated to telomere maintenance in many cases clearly has these kinds of effects. Okay, so causality in those clear Mendelian genetic mutation situations is very apparent. Now, what's interesting is that we learn another thing from genetics too, and that is that when genome-wide association studies were done, notably by Codd et al. A couple of years ago, when they looked at what kinds of alleles are quantitatively related to shorter telomeres in white blood cells as a kind of general systemic measure of telomere length. There's subtleties between cell types, but overall you get an overall idea of telomere maintenance. And what was found was that common alleles of, in some cases, some of the very same genes would have effects that were affecting telomere length. So these were rate limiting for telomere length. Now, the interesting thing was that in the population at large, this clearly could be related initially in initial studies to cardiovascular risks. And in fact, there were additive allele effects for just the 7 top scoring common alleles that were related to these telomerase and telomere genes, and a couple of others as well that clearly related to increased coronary artery risks. So this was looking very causal here. And similar findings have actually been found recently, same kind of approach, looking at these top scoring common alleles of these common telomere genes in increased risks of Alzheimer's. So this is starting to emerge as playing roles in these very important common diseases. Now, I want to emphasize right from the start, this is not the only thing going on at all in these common allele situations. Of course, there are many other factors that will play into this, but we're seeing contributory effects, and this is telling us about the biology. So, when we look at all these kinds of diseases that I mentioned were very characteristic of the population at large, and particularly with aging, when we ask what is involved in the etiology, well, we can say that there is some role that you can make a good case for a couple of them is really causally contributing, and in others look very like they're playing a role through a smoking gun kinds of evidence. So in general populations, this summary summarizes a great deal of evidence from many different cohort studies that telomere shortness, just as measured systemically by white blood cell telomeres average length, statistically predicts future diseases of aging, as well as being in a couple of these cases that I mentioned, actually having causal implications. So telomere shortness is something that statistically one can discern happening with age. This was with 100,000 people that was done with the Kaiser Permanente UCSF collaboration, and we measured telomeres in 100,000 people, and as a function of age you can see that the mean, which is a very tight mean, but the distribution is much wider. I'm just showing the error bars for the standard error of the mean, that goes down with age. But then interestingly, after the maximum mortality age, and this scale is off a bit, sorry, it should be around 70 to 75, you see the dip, but then people who live longer and longer seem to have a survival correlation with having longer telomeres, and there's good evidence that the telomeres had been longer through the rest of their lives, and that's associated with their increased longevity. We don't yet know the causality, but there's some reason to think that that might be contributing to some of the aspects of why people are surviving longer ages. So much to be studied there. But I just want to point out that there's a very interesting observations now about mortality in a couple of large studies, this large study of 100,000 people, where if people were in the bottom quartile of telomere length, where you can set the odds of mortality during the next 3 years, if you set the odds to 1, then you can see that the odds of mortality are reduced by about 20% in the other 3 quartiles of telomere length. Interestingly, that persisted after adjustment for multiple other mortality predictors, and I've listed them, obviously age, gender, race, ethnicity, education, cigarette pack years, physical activity, alcohol drinks per week, BMI, all of which themselves do show correlations with telomeres in the bad direction, but when you adjust for those, you don't shift that. Now, reassuringly, one sees the same kinds of things where mortality is also predicted by telomere length in a completely independent study, and I'd like to cite the Copenhagen study where Roth et al. Showed that, first of all, if you just look at raw data for telomere length, showing 10 deciles of telomere length, white blood cell telomere length, and ask for deaths in the next 7 years on average, you see a very kind of graded relationship. And in fact, that holds for a number of the major causes of death in this Copenhagen study as well, that there's this sort of graded probabilities. Now, this was unadjusted raw data. So the important question is what happens when you do multivariate adjustments, and indeed you still see a general gradation. It's down, but you still see a difference between the lower and upper measures of telomere length distributions. So there is this relationship. It is a predictor in a very statistical 40,000 foot up kind of view. So in general populations, I think we can safely say from these very large cohorts, completely different countries, a lot of wide ethnicities were covered in the California cohort, that there really is a risk relationship with shorter telomeres. Now, we can't say that this is causal, but the data I showed you before about the genetic data certainly implicate that some of this is actually contributing to the various factors that will go in and will ultimately, lead to the quantifiable mortality risk. But by no means is that the only factor, of course. But I think we can safely say that there is a relationship one can see. We know, of course, many factors play into the etiology of risks for these diseases. But we really can say that, I think it's fair to say that properly regulated chromosome end maintenance does help protect from these kinds of readouts, such as diseases and of aging particularly, and also mortality. And I think because they often go hand in hand, it makes sense that this can be one underlying system-wide reason that's contributing to this. So what about the kinds of effects that do have influence on telomere length? I've referred to allele effects, and they're quantifiable, they're subtle, but they're real. You can discern them in large enough cohort studies. But there's clearly non-genetic effects as well. And to summarize a great deal of research again, what we initially with UCSF collaborators showed, and then many others have shown, is that chronic psychological stress, and I'll give some examples, really has a clear relationship, as we know, of course, clinically, on disease risks and outcomes and so forth. And I'll give you one example in a moment. But also we find that that relationship holds for telomere maintenance in a whole variety of different settings that have a feature that you can capture in the very simplified term chronic psychological stress. And this means very severe generally. So what's been learned from a lot of observational studies is that, in fact, there are factors that are now not genetic factors per se, which can quantifiably affect telomere maintenance in dose-dependent ways in humans. And so the studies that I'll just summarize that relate to, how is the balance of telomeres overall, the long versus the short, the shortening versus the lengthening, how does this play out? when you measure telomere lengths, what does that relate to? It relates to, on the one hand, the kinds of factors that have now been well documented to be associated, and in some cases you can say causality, and are causing shortness of telomeres are those listed here. Chronic stress in various kinds of settings, such as chronic caregiver stress, caregivers of Alzheimer's family members, caregivers of their own children who are chronically ill. Depression shows real clear dose dependencies. Low education, interestingly, predicts shorter telomeres well into middle age and elderly. Prenatal stress, we see it in the childhood, in the telomeres. Childhood trauma categories, a whole list of things which, and then including lifestyle factors such as tobacco use, poor diets, obesity, even external situations such as environmental pollutants of various kinds, and also neighborhood disorder as being one way of capturing community sort of environmental issues. Conversely, there's also a list of factors which are important in terms of their positive association with longer telomeres, of which education level has tracked across large numbers of cohorts now, well into adulthood, and resiliency factors that can be captured under the headings of exercise and sleep patterns, and certain stress reduction interventions starting to show things. Omega-3 is the only kind of singled out item where I think you can say there's a relationship between telomere maintenance and that particular component. So really what's missing is, though, to understand how all of these things may interact with the small effects we see genetically, and these ones can have quite good-sized effects, but they're probably going to be very interactive with genetic factors. So I want to now just turn to something where you're saying, well, this is all fine and good, this is telling us about something with diseases, but how might this be clinically relevant? And I'd like to just give one example from a study that was done at MD Anderson. It was done with 441 bladder cancer patients, and what was done was to, at diagnosis, look at two factors at the time of diagnosis when they came in with the cancer, at the two factors which each had been independently suspected of having some statistical effect on what was going to be the prognosis for these bladder cancer patients. So I'm going to talk about mortality. So the telomere length and also depression, and these were quantifiably measured on standard scales and then just done as median splits for both. And what happens when you combine the two is that you greatly increase the precision of predicting who is going to be not surviving among bladder cancer patients. Measures are taken at the time of diagnosis when they've come into the clinic. Either one alone was not strongly predictive. So that's the conclusion of what I'm going to just go through the next few slides and show you. But the point being that I think often it's how factors interact. That's really important, and I think that's what the telomere measures are probably going to be much more useful for than just looking at them by themselves. So if you look at depression and you look at telomere shortness and you look at bladder cancer patient mortality, you see mild effect. There's some mild effect. I'll show you what that data looks like. If you look at them both together and ask who has both depression and shorter telomeres, both combined, that's a much bigger effect. So let me show you some of the raw data, the Kaplan-Meier curves here for the people who had one or other and then both of these, and these are the Kaplan-Meier survival curves. I'm going to show them for you in a different way, but I just want to emphasize this was after adjustment for all of the many factors that would have affected mortality, survival in these patients after diagnosis. And so even when you adjust for age, gender, ethnicity, smoking status, cancer stage, bladder cancer grade, and so on, still you see effects. So what I want to point out now is that same data, but just to make the point more graphically. So bladder cancer diagnosis, 441 patients come in, and they are stratified for whether they have depression on the high or the low end of the scale, median split, or they have short telomeres, again, longer or shorter in their white blood cells, or they have longer telomeres and no depression, that's neither, or they have both shorter telomeres and depression. So here we have 441 patients, all sorts of different people coming in. And now we look at 2.5 years and we say who died if they had neither of those two things, or just depression or just short telomeres. So a fraction of them have died after 2.5 years. But if you look at the people and look at the fraction who died after 2.5 years who had both depression and short telomeres at diagnosis, this is it. So big, you can see it already. But now if we go to the 5-year survival milestone, here are those, obviously more, who had neither or just short telomeres or just depression. And this is the fraction who had died. But at the 5-year survival milestone, if you had both, this is who had died. So it's very dramatic. So the point being that the two together are much more predictive, whereas one or the other is, not strongly predictive. Two together are very predictive. Now, of course, you step back and you say, well, this is actually precision medicine, right? Precision medicine is taking multiple factors and greatly increasing the precision of some measure. Often we think of it as treatment responsiveness and so forth, but, this is one kind of predictiveness. So in a way, it's an example of, quote, precision medicine at work, if you will. And I think there's real clinical implications here. It's an interaction of two factors, but there's, indications that then that would be useful information that would be clinically relevant because of these very big survival differences, despite all the adjustments for the usual factors that might be expected to affect survival. So that's the one case shown here. I think there's going to be very interesting possibilities to look at other diseases, not just telomere measures alone, but start looking at them in combination, led by this striking example. Now, this comes back then, though, I think, to something very important, which is the question that I raised at the beginning, which is how do we age? And in particular, how do we think about the bench to the bedside? So I've just told you humans have told us a lot about what goes on in aging of us. And that has been incredibly important because it now leads us into understanding the kinds of questions that we ought to be asking about these common diseases. So let me just say that aging is not just, telomeres. I've said it's not at all. And in fact, there's some interesting good things that happen as we age. Luckily, our brains do keep regenerating. And I just want to take an example, not all over the entire brain, but there are parts which regenerate. Neurogenesis goes on. One can take that into the lab and look at cells and understand, for example, disease. And so I just want to point out a really interesting case, in the case of autism, where this is a recent study where brains of people who have autism or not, about a third of them roughly, I think, had brains that are larger than expected for their developmental stage. And so when one takes simply skin cells from those individuals, differentiate them directly into neurons, as was done by Rusty Gage and colleagues at the Salk Institute, what they found was that those cells intrinsically grow faster, they proliferate faster, they make more processes. Even after you've taken them from skin and differentiated them in the lab, they recreate that characteristic that was found clinically in the autism patients by looking at brain scans, seeing their brains had somehow overgrown in some way. So what one's learned from this, by taking, this is the bedside, back to the bench, one has now understood something about what's going on in at least those kinds of classes of autism. And in turn, of course, this is about brain development. What does it mean when you have cells that are highly proliferative that shouldn't be? And this is intrinsic to these cells. So again, one is going to be able to learn about diseases such as autism and take that back from the bench again, back into understanding brain development and hence diseases of this. So similarly, the same kind of thing, one can grow kidney cells, from patient diseases. You can grow them in the lab and then learn how to bring that back into regenerative, hopefully regenerative clinical practice. But again, this is going to have to be a cycle. People at the Salk Institute work with this. Another really great example is the microbiome, of enormous interest, but you can learn how our immune system and how various components of immune cells are reacting to bacteria by understanding what goes on when bacteria, in this example, they actually cause cachexia, muscle wasting, and there's another bacterium that in the same person, and this was done in a mouse model but inferred in the same person, will actually set up a response in cells that prevents the muscle wasting. Now you know that, one can now think about drug-related approaches, which, by the way, don't involve antibiotics. So let me just finish with novel cancer treatments. And again, the bench led to cancer. Cancer took one into an understanding of cancer cells where it was learned that a common lung cancer tumor suppressor mutated gene in lung cancer was actually all related to energy and fat synthesis. And so one can now, from the cancer, human cancers, learned about a process that was very important in human cancers. And so turning that now again, that science, so we've got the cycle coming back again into bedside, very interestingly finding that an ACC inhibitor of acetylcholine ACC inhibitor in fat synthesis, when you combine it with standard chemotherapy, carboplatin in this case, actually gives you a remarkable reduction of tumors in 4 different models of lung cancer. So the point being that this cycle really plays out in different ways. So I just want to finish with the idea that, we really have to have new pushes in cancer. You've heard about the Moonshot Initiative, which was, the first thing after many decades in the US of pushing cancer when Nixon signed the NCI into its existence. And I was fortunate enough to be at this the World Economic Forum meeting where this was discussed in a panel, and I think the points that one can make in these kinds of settings are important. And indeed, I really was keen to make the point that you need this cycle continuously, not only of bench to bedside, but of bedside back to the bench, because this is the kind of cycle, I've given you a few examples, that I think can really push our understanding of disease. And so I thank you very much for your attention.
Thank you so much, Elizabeth, for that absolutely wonderful talk. We'll take questions at the end of the session if there's sufficient time. Our next speaker is Professor Dario Campagna, who's Director of Immunopathology and Cell Therapy at the National University Cancer Institute in Singapore. Professor Campagna's research career has focused on translational research in oncology. His laboratory works to understand the biological basis of leukemic cells and to translate his findings into new clinical treatments. He's won numerous awards, and he's also the scientific founder of 2 biotechnology companies. Please join me in welcoming Professor Campagna. The title of his talk is Cell Therapy of Cancer.
Thank you very much, Christina, and thank you to the organizers for the opportunity. It's an honor to be here. I'd like to discuss briefly how we can bring cells of the immune system to the clinic to treat cancer, and I think this provides a pretty good example of bench-to-bedside medicine. Let me tell you a bit more about the basic of the science behind this technology. We can activate immune cells in vivo by administering antibodies that release immune cells from suppressive mechanisms that tumors can exert. We can also give bispecific antibodies that attract immune cells to the tumor site. My interest is more on the direct administration of immune cells, and when I talk about immune cells, I specifically refer to T lymphocytes and natural killer cells. So the idea here is that we can take immune cells from a patient or a donor and then manipulate them ex vivo to make them specific against cancer cells. And the basic concept is that ex vivo we can introduce a receptor that would allow immune cells to recognize molecules expressed on cancer cells, and upon contact the immune cells will become activated and exert cell killing. And this concept was in fact initially introduced in the late 80s early 90s, by an Israeli scientist called Zelig Eshar. And Dr. Eshar took a portion of an antibody, the part of the antibody that binds directly to the antigen, and used it to compose a chimeric molecule. One of the components is this portion of the antibody. Another component is a peptide that anchors this portion of the antibody to the cell membrane. And finally, there is a signaling cassette that allows this artificial receptor to signal. So this is the basic structure of what we call today chimeric antigen receptor. And I think it should be underscored that when Dr. Eshar was doing this research, his initial motive was entirely basic research. He was interested in seeing how signal transduction will occur in immune cells. So there was no really translational intent in the initiation of these experiments. So this is what is the receptor that Dr. Eshar developed is what is now called first generation chimeric antigen receptor, or CAR. And we and others felt that this receptor, although very exciting and interesting, had somewhat a diminished stimulatory potential. And we knew from basic immunology research that the single stimulus that this receptor had was not sufficient to induce a durable and potent T-cell stimulation. So we and others added another component to this receptor, which is a co-stimulatory molecule. It's an additional stimulus that allows T lymphocytes to proliferate and expand and exert a more powerful anti-tumor activity. So this is now called second-generation CAR. So two signaling molecules, one is a primary signal and the other is the co-stimulus. And the other important point about CARs that needed to be resolved in terms of methodology is to identify a good target expressed on tumor cells that we can target with our receptor. And this is not such an easy concept because most molecules expressed by tumor cells are also expressed by normal cells. There are very few molecules that are expressed exclusively or nearly exclusively by cancer cells and are accessible on the cell membrane. At that time, we and others selected a molecule called CD19, which is expressed in all B-cell malignancies, acute lymphoblastic leukemia, chronic lymphocytic leukemia, non-Hodgkin lymphoma, and among normal tissues is expressed only by B lymphocytes. And B lymphocytes are dispensable cells. We can live without B lymphocytes for a while. So the technology is quite simple. We can take blood from a patient by leukapheresis typically, so we can obtain a large number of white cells. And then these white cells are manipulated in the lab where they are genetically modified by expressing this receptor, usually using viral vectors to introduce this molecule into the cell. And then these cells are returned to the patient after about 10 days of manipulation ex vivo. So this relatively simple manipulation and this relatively simple receptor, when actually used in clinical studies, resulted in phenomenal responses. And the very first paper was published in 2010, a single patient with B-cell lymphoma by Dr. Rosenberg at the NCI, and then subsequently in 2011, the group at UPenn published dramatic responses in 3 patients with chronic lymphocytic leukemia that received these T cells armed with this chimeric antigen receptor targeting CD19. And since then, several other groups have reported quite dramatic successes after infusion of these T cells. And just to give you an idea of the responses that can be achieved in acute lymphoblastic leukemia, which, by the way, is the most common form of cancer in children, this table summarizes the results that have been achieved so far. And as you can see there, out of 153 patients enrolled at various centers, 87% achieve complete remission. And of these, 88% achieve molecular remission. Molecular remission means that you cannot detect leukemic cells in the bone marrow or in the peripheral blood with a sensitivity of 1 in 10,000 or more. So really these are phenomenal responses, considering that these patients were patients that have relapsed multiple times and they were completely resistant to very intensive chemotherapy. So quite an astonishing response, and the responses in many of these patients have been durable as well. So these publications, starting, as I said, from 2010, 2011, attracted a great deal of attention, also from the lay press, as you can see in this slide. And suddenly there has been a very vigorous interest on the part of pharmaceutical companies and investors in this technology, really motivated by these successes in the clinic. So one needs to keep in mind that before 2011 or 2010, nobody was interested in this technology. Scientists, physicians interested in this field were really a very small group of individuals, and certainly pharmaceutical companies and investors had zero interest on this. So things have really changed dramatically over the last 5 years. So if we look at the history of this CAR technology, as I said, Eshar developed his receptors in the late 80s, early 90s, and it took about 20 years for this to be commercialized, or at least to be recognized as a potential drug to treat cancer in the future. So this seems a long time. But in general, there is a resistance to novel ideas, not only in the medical community, but in general. So if we look at how long it took us to put the wheels in the right place on a suitcase, it took us 37 years. So it really confirms that it's not easy to introduce innovation in medicine. Of course, once the proof of concept is shown, then variations on that concept are easy to introduce, but really the main initial changes take a long, long time. So if one is interested in translational research, I think he needs to be prepared for a long haul. So in addition to modify T cells with CARs, there are many other ways in which we can modify them. We have been working on a technology that allows T cells to recognize a portion of an antibody. So in this case, we can genetically modify T cells with one receptor and then use any antibody available in the clinic to redirect these T cells against different types of tumors. You can also modify T cells with T cell receptors. And then there is another cell type that we can also tap for immune cell therapy of cancer, and these are natural killer cells, and there is a lot of technical details that I cannot go into, but this, I think, is another very promising technology that is worth translating to the clinic and testing it, and there are studies ongoing in that regard. So traditionally, I think the translation of discoveries from the basic research to the clinic need the intervention from pharmaceutical companies and biotechnologies. But I think in this field, particularly in the field of cell therapy of cancer, there is room for a direct academic translation. We can take these therapies, make them ourselves in our hospitals or universities and do initial clinical trials. And this in turn may attract attention for pharmaceutical companies and investors and complete the circle. So to this end, at the National University of Singapore, we're fortunate to have a lab that allows us to process cells for infusion. So this cannot be done in any research lab. You need to have a special lab that complies to international so-called good manufacturing practices regulations. And unless you have that, obviously this is a non-starter. If you don't have that kind of laboratory, it's very difficult to do this type of work. It's impossible, in fact. And it's not easy to have such a lab because this lab financially normally is on the losing side, and universities are not particularly keen in maintaining such a structure. So it's always a struggle, but anyway, we have it and we are using it. So based on that, we have initiated a number of clinical trials exploring some of these newer ways to genetically modify T cells and NK cells, and the trials that we have ongoing at various institutions in Singapore are shown here in this slide. So I think the determinants of progress in this field, which again I think has provided a very good example of how we can translate discoveries from the lab to the clinic, is really three ways. One, we have to increase knowledge and understanding how the immune system works, because By knowing that, we can improve our ways to modify immune cells and make them more and more potent. And then we have to improve methods for large-scale GMP-grade cell expansion and genetic modification. At the moment, this is done in a very crude way. It's really standard tissue culture, as we have been doing in the lab for 3 decades. So there is a lot of scope for automation, robotization, and I think this will also reduce the cost of this type of therapies. And then there is a need for improvement in clinical trial design, because the current eligibility criteria and response criteria that we use are those really that we use for chemotherapy or for radiotherapy that are not really adequate to evaluate the activity of this newer therapy. So there is a lot of work needed in that field too. So one last slide is about the need of commercialization of this technology. So we know that, continuing the analogy of the car, we can do this in an academic setting. We can make effective receptors, we can make effective cells that can induce remission in patients with leukemia, lymphoma, and hopefully also solid tumors. But this can be done in a very limited number of patients. We are not reaching really the vast majority of patients that really would need this type of therapies. And also the product in itself is not particularly sophisticated. So we need to improve in that. We need to transform these little almost pedal-like cars that we are making into very powerful automobiles, perhaps in kind of off-the-shelf vehicles that we can just administer on demand to patients. So this requires commercialization. And to this end, we have been pursuing this route, and so we have licensed some of our patents to Juno Therapeutics. And as Christina said earlier on, we started 2 companies that plan to explore some of the technologies that have been developed in our lab. So this is my last slide, and I thank you for your attention.
Thank you so much. That was a wonderful talk. I've never understood CAR T cells so well. Our next speaker is Professor Angelika Eckert, Director of the Department of Pediatrics at the Division of Oncology and Hematology, and the Director of the Comprehensive Cancer Center at the Charité Medical University here in Berlin. Professor Eckert holds an Einstein Professorship, and her work is on the molecular biology and genetics of neuroblastoma, the 3rd most common solid tumour of childhood. The title of her talk is Personalised Medicine Approaches to Pediatric Oncology.
Thank you very much for the kind introduction, and also thanks to the organizers for their invitation to actively participate in this great meeting. It is truly a pleasure. So today I would like to share with you some examples from academic translational research that can contribute to the value chain, ultimately leading to personalized medicine approaches, in this case in the field of pediatric cancer. So we always see that it seems to be very simple. Cancer is in the primary focus of personalized medicine approaches, and we find figures like that where we take out the tumor of a patient, find the right DNA mutations, then it's very easy to find the right drug. Maybe we test it in parallel in animal models, and then we come up with the right treatment approach and we cure the patient with just one single molecular targeted drug. So it looks very simple, but is it really that simple? A couple of years ago, I talked to a systems biologist, and he told me, well, you know, if the medical people just would do it in the same way as the physicists do, using a lot of computer power to understand very complex things, then medicine would approach much faster in the right direction. And the physicists, they can understand something as complex as an airplane. And I thought by myself how very naive this kind of thinking is, because the complexity of an airplane, even compared to a virtual patient, is nothing, and it's not even the same dimension as the toy of a 3-year-old compared to a very complex puzzle. So it's definitely not that simple. It's a lot of work, and we need a lot of approaches, a lot of data, and a lot of expertise to really get this to a successful story. So for personalized medicine approaches, we need a very complex data pyramid, and we have to collect all this data in hard work. It's patient data, clinical data, but then the next level, of course, molecular data that we have to obtain from the biomaterial of the patients. It's sophisticated bioinformatics approaches, and then we have to check the utility, and finally we might get all the way to applications. And what we definitely need for this is an interdisciplinary translational expertise of all subjects that can contribute to this, and definitely clinical scientists. So after all, a capable and productive university medicine that is able to provide this clinical expertise to it. So in the field of applications, I would now like to share 3 examples with you showing how this has been approached in the field of pediatric cancer. Pediatric cancer is in general considered to be an excellent model for molecular diagnostics, as all the genomes that we find in pediatric cancer entities are quite simple in comparison to adult tumor entities. For example, melanoma or lung cancer are quite difficult to understand, whereas pediatric cancer in the genetic setup is so simple that it's really a useful model. Also, there are internationally a lot of activities, and excellent genomics and transcriptomics data have been collected over time. We share in Europe a combined molecular database at the University of Amsterdam, where we provide all the molecular data sets of that have been characterizing pediatric cancers, and we take advantage of this. And after all, in the past years, we had the problem that personalized cancer medicine in pediatrics was considered to be difficult, but today rare, as in pediatric cancer, is the new common, so we can really learn of pediatric cancer models. Also in pediatric cancer, there's internationally an excellent shared infrastructure around. There are dedicated clinical centers treating patients with childhood cancer, and usually there are national biobanks that collect the biomaterial, for example, here in Germany, of more than 90% of the patients in one central biobank in the country. We also share national clinical databases, and we have national trial coordination centers telling the whole country what to do, what kind of treatment protocols to use. And in pediatric oncology, we like to obey these rules, and we contribute and treat all our patients according to the same algorithms. We are meanwhile also in the process of sharing international databases. This is an example of neuroblastoma, which is my field of expertise. So we are currently setting up at the University of Chicago an international neuroblastoma database. It holds already more than 11,000 patient data sets from the clinical side, and we are currently in the process of enriching the database with molecular data sets. So let me share some examples from the field of neuroblastoma with you. Neuroblastoma is a common childhood tumor of the sympathetic nervous system, so that explains why it's localized in the adrenals and in the sympathetic chain. It counts to 10 to 15% of childhood cancer cases. It metastasizes to the bone marrow and liver, and most of the times this is already there at the time of diagnosis. And we observe an extreme clinical and biological heterogeneity. So if we look at the patients, we really have the same name, neuroblastoma. The pathologist cannot tell us apart, but it's really different diseases. You don't have to be an experienced pediatric oncologist to tell that there is a difference between the child on the left and on the right. The child on the left is suffering from a stage 1 neuroblastoma. The survival is excellent, more than 90%. And we observe a very amazing biological characteristic in these patients. They show spontaneous regression without any treatment. On the other hand, half of the patients show up as metastasized disease, stage 4. So on the right, this is not a better child, but it's suffering from bone metastasis to the orbiter. And the survival in these children, no matter what we do, very complex chemotherapy, irradiation, stem cell transplantation, differentiation therapy, immunotherapy, the survival is still less than 40%, and we have to solve the clinical problem here. And it's not a matter of time that the one develops to the other, but they are very different in their biology from the very beginning. And of course, it looks like a very simple picture, and it looks like we don't know any molecular diagnostics to discriminate between these two children. But of course, it's not always as simple, and we have cases that are in their clinical appearance somewhere in between, and there, of course, molecular diagnostics comes into the game. So the first part of personalized medicine applications is in the field of risk stratification. So we need to decide which patients need treatment and how much they need, so not to endanger them by undertreatment or by overtreatment, because by their clinical appearance we cannot precisely judge this. Again, this work is a lot of work and it takes time, similar to the field of CAR T-cell therapy. So in the German neuroblastoma community, we started in 2005 with different molecular profiling approaches, and 10 years work was focused on defining reliable molecular outcome predictors. We played around with a lot of technological omics platforms, with a lot of sophisticated bioinformatics approaches. And now in this year, finally, we came up with a prognostic classifier based on mRNA expression that is now ready for treatment stratification within routine clinical applications. So this classifier has been analyzed first in an initial cohort to identify the genes that are relevant for prediction. Then it has been validated in a retrospective tumor cohort, and then in a prospectively collected tumor cohort within a clinical trial. And this is certainly the robust way to go if we really want to rely on this data. So by this classifier, we can meanwhile, with an accuracy of 0.95%, we can determine if these patients will have a favorable outcome or an unfavorable outcome, and decide to either observe them only or to give them aggressive chemotherapy. So this is ready for clinical application. The second field of personalized medicine approaches is in the identification of targetable genetic alterations, because we don't only want to tell the patients apart, but we want to solve the clinical problem for the aggressive subgroup of patients. So we learned in the past couple of years a lot about the genetic landscape of neuroblastoma in this wonderful paper of our American colleagues, and we realized that there is only a handful of mutations characterizing the tumor samples. By a second look into these data sets, we realized that TERT rearrangements, so again the telomerase field that we have learned so much about today, is also very significantly changed in neuroblastoma, giving us new insights into the pathogenesis of the disease, but also having a significant impact on the outcome of the patients. But then we thought, well, we have the picture, but we decided to analyze also relapse samples, because we need to know what is happening when a tumor is recurrent, and we came to a completely different picture. So we found new mutations in the relapse tumor in comparison to the primary tumor, and only a few recurrent mutations in this sample set, but everything that you see here in red are new mutations that have not been present in the first tumor. A part of them was selected, being there in very single clones, not detectable in the primary tumor, but then selected to a larger extent in the relapse tumor. But we have to conclude that the tumor indeed is learning and adapting to therapy, coming up with new mutations, and it's not sufficient to have an initial biopsy of a primary tumor if we really want to treat the patient based on molecular diagnostic information. This has to be from a relapse biopsy. And in this field is my last example. So we have the problem of intratumoral heterogeneity that we have not solved yet, and this can occur both in spatial ways, so all the metastases are differently composed in comparison to the primary tumors, but also in a temporal way. Temporal heterogeneity means that there is a development from the primary tumor to the relapse tumor. So although we have not really a good solution for this intratumor heterogeneity, we started now to do an ultimate test of personalized medicine by applying molecular diagnostics to tumor samples. And of course, you have to do that within clinical trials. And finally, to really prove that personalized therapy brings a benefit to the patients, because that not in every field has been shown today, although we have high hopes. So we asked the question how to improve the cure rates of relapsed pediatric cancers, and selected the 11 cancers with a cure rate of less than 10% in the relapse situation to see if there was a benefit of personalized medicine trials. We teamed up all over Germany under the roof of a project that we call INFORM, Individualized Therapy for Relapsed Malignancies in Childhood, and we use this as a blueprint for personalized medicine trials also in the German Cancer Consortium. 58 clinical centers in Germany are contributing to this trial, which is coordinated by distinguished colleagues from the DKFZ, Stefan Pfister, Peter Lichter, Olaf Witt, and by myself. And the recruitment of the patients is running very smoothly. so that we were able to set up a registry phase within 2 years to establish all the workflows. The samples from every German center are sent to a central pathology lab at the DKFZ. Then a complete molecular profiling platform is applied to the samples. Sophisticated bioinformatics analysis are extracting the information. The 5 most important targets are validated, and then the information is given to a molecular tumor board where it is discussed, and according to very complex algorithms, the targets are prioritized according to their potential clinical benefit. And the workaround of these platform, meanwhile, is 21 days between sending in the samples and delivering the complete information. So the next step now will be to really get into a clinical trial where we use combination treatment of molecular targeted therapies in 10 different arms based on the targets that we have identified in the 2 years registry period, so that we know what kind of targets are there very common, in what combinations they occur, and now they can be targeted by treatment. And you can imagine this is new also to pharma industry, to combine drug A from pharma company A with drug B from pharma company B needs some discussion and needs some rethinking of the field. So we hope that we will get that done and open the trial in 2017. And a lot of European countries have already expressed their interest to join forces to recruit patients to this trial, so that we hopefully can finally show that there is a benefit of molecular targeted therapy for these subsets of patients. This is my final slide. So in Berlin, we decided to define a complete personalized medicine cycle, and we are happy that we are supported in this research endeavor in a collaborative research grant of the Berlin Institute of Health. We call it Terminate NB, with an ambitious goal, of course. And in this cycle, we want to understand from tumor biology on the left, applying for early diagnosis all imaging qualities that we have, but also, of course, histology and including liquid biopsies of the patients. Use these biosamples for a complete panomics technology platform, give the data to our bioinformaticians for network analysis to extract the key molecules from passenger mutations that are not that important, to validate that on a platform in silico, in vitro, and also in vivo models. Then apply the new knowledge within clinical trials and use these clinical trials, the responders and the non-responders, to learn what is going on and why they are responding based on the biology of the tumors. So by this, I'm finished, and I thank you for your attention. And translational research is always a team approach, so I also thank the team in Berlin and the team of Inform that is doing a great job in applying translational research to biosamples of pediatric cancer patients. Thank you very much.
Thank you very much for that absolutely brilliant talk on the complexities and the massive workflow that's required to have a personalised medicine approach. So our next speaker is my colleague from Monash University, Professor Stephen Jane, who's head of the Central Clinical School and Director of Research at Monash University and Alfred Hospital in Melbourne. Steve is a haematologist who works on the genetic basis of developmental and acquired disorders of both the blood and the skin. In January of 2016, he had success with commercialising a discovery, and a suite of drugs were developed by him based on his discoveries and have been licensed to the global health leader Merck Sharp and Dohme in what is believed to be one of the largest preclinical licensing deals in Australian biotech history. So he's here to tell you his story. He's halfway through, or almost past, the Valley of Death, and the title of his talk is Nullable Therapies for Hemoglobinopathies.
Slide. Oh, thank you. Right, thank you very much to the organisers for the invitation. So I have been working for many years on 2 diseases which are certainly not out of place at the World Health Summit, namely thalassemia and sickle cell disease. So these are the commonest genetic diseases worldwide, and their prevalence equates to the coexistence of disease alleles where malaria is endemic. So consequently, they're prevalent throughout the Africa and Asia, North and South America and the Mediterranean basin. And obviously with migration patterns, this has changed substantially, particularly in North America and more recently in Australia. About 10% of the world population carries a globin chain disorder, which I think is a mind-blowing figure with any disease allele. So 1 in every 10 human beings on the planet has a globin chain mutation. They're devastating disorders to look after clinically. Beta thalassemia results from null mutations to the beta globin gene, part of the hemoglobin molecule, and children require lifelong blood transfusion for survival, and that's fine if you live in a Western nation where that is available to you, but unfortunately where this disease is endemic, many of those nations do not have safe or adequate blood supplies, and so consequently the life expectancy in that consequence is only 4 years. But even in the best health centres in the world, life expectancy is only 50 years, predominantly related to consequences of chronic transfusion and iron overload. Sickle cell disease is probably even more devastating as a disorder. It is a point mutation resulting in an amino acid change to the beta globin molecule, which results in an inability of the globin molecule to be modified as it transverses through the microcirculation in red blood cells. And so the red blood cells get trapped inside that circulation and block, leading to downstream ischemia and organ infarction. And kids with this disorder really have a miserable existence. They have problems, obviously, with anemia, but also infection, incredibly painful vaso-occlusive crises, and downstream organ effects, including blindness, renal failure, psychosocial issues, and ulcers, etc. A really devastating disorder, and untreated, only 14 years of life expectancy, and again, even in the best centres, life expectancy only 42 years. Paradoxically, we've known how to cure these disorders for many, many years. So the above part of that slide is the beta globin cluster, and there are a number of genes in this cluster that are expressed developmentally, and they're arranged on chromosome 11 in the order in which they're expressed. So initially in the first 5 to 6 weeks of gestation, the fetus survives on embryonic hemoglobin, and then the first of the hemoglobin switches occurs where we move from embryonic to fetal hemoglobin. And even children with beta thalassemia and sickle cell disease exhibit no symptoms in utero because the fetal hemoglobin is protective. And it's only at birth, when the fetal to adult globin switch occurs, that the diseases begin their onset. And essentially, as fetal hemoglobin goes to 0 and adult hemoglobin rises, obviously in beta thal you never make the adult hemoglobin and the disease manifests, or in sickle cell you make the abnormal form. But amazingly, there's been a very, very small cohort of patients with those disorders in the homozygous state who inherit another mutation that instead of their fetal hemoglobin switching off at birth, it remains on, as shown in the green line, and those patients, even with elevations of fetal hemoglobin to only 20%, are virtually cured. So nature's put down the gauntlet, said, here's the challenge. You can cure these disorders if you can reactivate fetal hemoglobin in all the patients that don't have a mutation to do it for them. And so our lab and many other labs around the world have been really working hard to try and understand what is the molecular basis of fetal hemoglobin gene silencing, with a view to targeting that, reversing it, and then instituting a therapy in the worldwide number of patients with thal and sickle cell disease. So the problem is that largely we're trying to drug the undruggable. And for those of us who work in the idea of developing therapies, transcription factors are the undruggable. I've had many conversations with molecular medicinal chemists talking about developing inhibitors to transcription factors, and they're very brief conversations, I can tell you, because they're very, very difficult molecules to access and to target effectively. And so what we need were different strategies here, and I always love showing this slide because essentially in one particular pictorial it summarises about 15 years of work by probably hundreds of people. But essentially, through just dogged persistence really, the lab eventually identified something that gave some interest to the medicinal chemists, and that part of a transcription factor conglomerate that's important for silencing fetal hemoglobin, one key molecule that is recruited into this process is shown in the bottom, and that is a molecule called PRMT5, which is protein arginine methyltransferase 5, which is an enzyme. And it's a critical enzyme for epigenetic silencing of fetal hemoglobin. So many human genes are silenced epigenetically, and that is that there are no changes to the DNA per se, but the proteins that adhere to the DNA are modified, and that changes the transcriptional state of that particular gene. And PRMT5 is a linchpin in this process. It recruits a whole raft of epigenetic modifiers, including ones that actually modify the DNA itself by methylating it. And it's long been known that DNA methylation and fetal hemoglobin silencing are intrinsically linked. And so we've found out that by knocking down PRMT5 using a genetic approach, that we can undo all of these epigenetic locks and chains and now liberate fetal hemoglobin to be re-expressed. And because it's an enzyme, that gives us an opportunity to develop target drugs that might block its enzymatic activity. And so when I finally went back to the medicinal chemists with this idea, I actually got a seat and a cup of coffee, and they were interested in developing the project. So these are the drug development pathways that we face, and Chris talked about the valley of death, and that's the journey from target discovery all the way through to a molecule that might be efficacious. And this really is a sobering thought when you look at this particular diagram. So within the drug discovery setup, we start off with potentially 10,000 compounds, and by various stages of attrition, which relate to failed preclinical development, toxicology, bioavailability, and so on, at the end of this funnel, with 10,000 starting compounds, we come out with one drug. So there are very many hurdles to fall at in this journey. So just briefly to describe our pathway, which I think is a good pathway to hear about because it really shows many of the difficulties that we face, not only in Australia, but I think worldwide. So our initial problem was funding, and particularly all of our target discovery work was really cobbled together with a raft of funding agencies, including the Australian government through the NHMRC, which is our MRC equivalent. We got a grant from Roche to develop this, and so on. And so much of the early work was funded through that process. But then the roadblock really starts, is that when you have potential molecules that could go further, how do you get those all the way down the clinical pipeline? We had some interest from venture capitalists, but the problem in that situation is they want 99% of your intellectual property and give you 1% of the funding. And so that really wasn't a very appealing approach to us initially. And so we really continued to try and nickel and dime and thread together monies together to get us further down the path that would then make us competitive for bigger stakeholders to be engaged. So the way we did this, we set up an assay using one of the proteins that PRMT5 modifies, a histone, and then we screened a library through an academic collaboration with the Walter Eliza Hall in Melbourne, and we identified a number of potential molecules. Amazingly enough, when we looked at these, they had incredibly high specificity for the target that we were looking at. So most of the other enzymes involved in epigenetic modification were untouched by these particular drugs. And the reason this just shows a Western blot showing a complete eradication of the mark you can see in the top two panels induced by this particular enzyme. And the reason these drugs were so specific, we found out from doing structural biology, and that they don't bind in the enzymatic pocket where there are a lot of family members that might be inhibited. They actually bind in the substrate binding site. And so you could specifically inhibit the activity of this particular enzyme with a compound. So there was only opportunity for drug development through cancer in Australia, and the government had set up a cancer research consortium to develop small molecule inhibitors in the setting of cancer. They had no interest in looking at thalassemia and sickle cell disease, and this is a general problem that we face, that orphan diseases such as sickle cell and thal, despite their worldwide prevalence, have to date had relatively modest interest from pharmaceutical industry because of the distribution worldwide. Fortunately for us, despite the fact that initially there was not much on PRMT5 from a myriad of labs around the world, data was emerging that PRMT5 actually seemed to be an important molecule in cancer, and this is pretty much summarised on this slide. And it's actually true for many epigenetic modifiers that they seem to play a role in basic biological processes such as globin gene regulation, but they also have key roles when perturbed in cancer formation through a myriad of mechanisms. And so we went back and knocked on the door of the CRC for Cancer Therapeutics with all the information on cancer, and then they agreed to take our target molecule into development for both cancer and sickle cell disease and thalassemia. So what they brought to bear was a huge number of medicinal chemists. We had, I think, 12 med chemists working full-time around the clock on improving the original molecules that we brought to them as inhibitors, because obviously from discovery through to drug is a huge pathway of iterative cycles of improving bioavailability, toxicity and so on. And so with that workforce we were able to generate lead compounds that looked as though they could go on to be drugs. But we're still in the valley of death here, an enormous step forward from just having potential drugs through to actually getting into clinical trial. And at this point, we approached the Wellcome Trust, which is one of the few organisations in the world that does have a funding pipeline for this in a competitive process, and we were fortunate enough to be awarded a large grant from the Wellcome, around £4 million, to try and develop our initial drug discovery through to clinical trials. And so this included scale-up GMP synthesis, as Dario mentioned, and eventually going through into clinical trials in patients with sickle cell disease in the first instance. We've continued to work on the cancer angle, and one breakthrough from our laboratory that I think has made this even more interesting is that these inhibitors seem to be extremely important in the development of blood cancers, and in particular acute myeloid leukemia. So on the left panel you can see the expression of PRMT5 in normal bone marrow, and in the bottom you can see in brown the expression in acute myeloid leukemia, incredibly high expression levels. And fortunately for us, that when you treat either normal bone marrow stem cells or leukemic stem cells with this drug, we see a vast differential effect. So the normal stem cells are virtually untouched, whereas the leukemic stem cells are essentially eradicated. And we've now gone into animal models to show we can now at least cure mice with acute myeloid leukemia using these anti-PRMT5 inhibitors. So at that point, we really had a suite of drugs with great bioavailability, limited toxicity, activity in sickle cell disease and thalassemia, and also in blood cancers. And we had a suite of suitors who knocked on the door, and eventually a deal was signed with Merck, Sharp and Dohme that was summarised in Nature Review's Drug Discoveries early this year. And the licensing agreement is of the order of $500 million in milestone payments plus royalties. And most of this is going to go back into the CRC for drug development and to the university, into the Monash University program. And so there will be a reinvestment, I suppose, to look at new compounds, new drugs and new targets in different disease states that the university will be able to sponsor. So in summary, we have a first-in-class drug for potential treatment dear to my heart for sickle cell disease and thalassemia, but also potentially in blood cancers. And these PRMT5 inhibitors show all the properties that you would like of a drug in terms of toxicity, bioavailability and efficacy. And hopefully we still have a journey to go. And I think this is probably a sobering slide that we're about halfway from bench to bedside, which puts us at the bottom of the canyon, but hopefully we'll get through the pipeline and these drugs will come into clinical utility. Thanks.
Thanks so much, Steve. That fabulous story of your own personal journey. Our final speaker of this session is Professor Daniel Ford, who is Director of the Johns Hopkins Institute for Clinical and Translational Research in Baltimore. For the last 7 years, he's been Vice Dean for Clinical Investigation, a significant role at Johns Hopkins that tries to help others navigate their way through this complex and dangerous pathway into research translation. Thank you.
Great, thank you. Well, again, I'd like to thank the organizers for inviting me to come and participate in this interesting forum. Let's see if we can get my slides. There we go. So I just want to talk a little bit, because I'm going to be talking about the partnerships between academic and commercial, and just remind you a little bit of the very limited amount of work I've done with pharmaceutical companies, and also my experience with licensing a product. So in the United States, and I think this reflects really a worldwide movement, talking about universities becoming more focused on translational research. And our institute at Johns Hopkins, and just to remind you, Johns Hopkins, a very large, comprehensive university with a medical school, a school of public health, a school of nursing, lots of resources to bring to bear in this area. And we have created, with funding from the NIH and others, the Institute of Clinical and Translational Research. And I think you see our mission being much about what's been discussed today, about how do we do more efficient, faster, and optimizing this delivery of going from basic discovery to products that improve human health. And recognizing that we also have to work with the broad community, our local and national, whatever we identify as our patient community, to create interventions that are going to be important to them. We've talked a little bit about the translational pathway. I think you see it here described in one of the very linear figures, which kind of goes from basic discovery, mechanistic studies, initial human testing, proof of efficacy, effectiveness, diffusion in all healthcare settings. This sometimes seems more applicable to the drug discovery paradigm, which still remains the most common paradigm, easiest in some ways for us to understand. You notice these arrows back to basic discovery from each phase, which is where I think those of us that lead universities need to make sure this cyclical process occurs and recognize that, in fact, universities frequently have this unique combination of basic clinical investigators, clinical patients, that allows us to move through this cycle more quickly. I think you see in several of these examples, too, that a basic discovery leads to some, your first area is one specific disease, and then as you learn more, you understand it may be applicable to other diseases. That's where universities should really be particularly good at moving these ideas in a quick and efficient way from one area to another. This is another way to describe the translational research process, which is a much more interactive, saying that it isn't quite so linear. And I know many of you are here from the public health field. I mean, when you go through examples of translation, some of them don't follow this very linear pathway at all. I mean, one that I know we have looked through, even the position of sleeping for infants. It started with a thought, an epidemiological discovery. You move to a large public health campaign, and health was improved. It didn't have to go through the same regulatory linear process to make a difference. Now, when we talk about translation, though, inevitably it gets to this question about academic entrepreneurship and how do we create the environment that's going to efficiently allow those in the academic sector to work in the commercial sector. And it's great. I mean, you see some of the people up here, the enthusiasm you get to take a discovery through to a drug that is widely used. You don't want to lose that enthusiasm. But it's hard. Many times you'd fail. It's a long-term process. And we have to figure out how we're going to support faculty in that process. But usually when we think in this academic entrepreneurship, there's three different ways, right? One, conducting research funded by companies with the generation of new intellectual property, which is usually under some control of the commercial partner. Then there's the whole patents and licensing of intellectual property. And finally, this creating startup companies, which is clearly a phenomena in the United States, but as we see also true in Singapore. So why has this happened? Well, there is broad pressure on universities to increase commercialization. The politicians and others that fund research expect university-based research to have significant local impact, frequently talking about creating jobs, which is in some ways kind of the last phase. University activity and technology transfer is clearly increasing. Now, Johns Hopkins has historically been thought to be not one of the most active in this area, but I think it's true to say in general that most universities are still going to be getting less than 10% of their research funding from this effort. Entrepreneurship training is increasing in biomedical researchers. We have many programs now that are taking faculty members through a process that says, I think I have an idea that might work. I'm going to help you partner with entrepreneurs, business, and others through a process that helps them decide if this is where they want to go and how they can get there more efficiently. And then, of course, the reality is in many places that government funding for research is relatively flat, and so the average faculty member is saying, well, where else can I go for funding to help me in the progress of my ideas? So I don't want to say that there are no benefits to this, because I definitely think there are, but I also want us to look at the other side. You know, it is true that in many cases when you say, I have discovered a pathway, I have a target drug, I want to get it broadly to the global population, you have to go through some commercialization process to get there. So in many cases, it just makes sense in that pathway to having a broad impact. It clearly does bring another source of funding to support research in universities, and diversifying that portfolio is important. And I guess it encourages faculty to learn about the market and how to create innovations that more rapidly improve health. We think that it does help change the mindset of the investigators that says, you know, gee, I need to understand the process. And at the end of the day, for me, a translational researcher is somebody that is always thinking of the next phase and has a target in mind. They have a goal in mind that they can see how they would get there. Now, many universities have tech transfer offices, and this is the Johns Hopkins mission statement, and I think you would see most of those. We help inventors and entrepreneurs protect and license their innovation. Our mission is to protect the intellectual property of Johns Hopkins faculty and to commercialize these inventions for the public good and bring the benefits of discovery to the world. A very laudable goal. I think that's where we want to go, but there's a lot of decisions that need to be made along that process. So what's the public view of university and commercial collaboration? And surprisingly, I had a hard time finding much data on exactly how the public sees this. And I turned to really a well-done study in Canada that I'll tell you a little bit more about. But I think these give you some of how the public sees it, right? Yes, public confidence in research is reduced to some extent by involvement of industry. More concern about, are the results objective? Do I understand the biases? On the other hand, they clearly have high expectations for medical benefits from scientific advances, moderate benefits that it's going to impact on the economy, but they understand that a role for commercialization is there. This is based primarily on a study done of 1,000 individuals in Canada. And interestingly, they didn't really get to answer the questions until they passed a test that demonstrated they really understood these complex concepts. They saw the benefits of more rapid practical application, that they were more concerned about the issue that if it makes sense, an academic and a university works on an issue and commercialization is the next step, that's good. Where they become most concerned is if universities pick the problems they work on or pick the solutions to those problems because they believe it has a commercial potential. So, I mean, it does kind of surprise me working in the behavioral area for some time, right? We know there are lots of ways that we can promote behavioral change for healthy behavior of exercise and others. As soon as the app world becomes part of the community and people think, well, I can make money through apps to generate this behavior change, all of a sudden we have everybody working on apps. Yet I think if you looked at all the ways we could change behavior, the apps would not come up as the top way to achieve that. So I think those are the issues that we need to think about on the university side when we pick how we're going to move ahead in commercialization. And I do point out that this is, a moderately low response rate, but from the American Association for Advancing Science members, who actually, if you look, are now saying they believe there's too much pressure to develop marketable products in universities. So, and then imagine just the university of the future. Would we promote a faculty member? Would they have a faculty member in, would they have a career in academic medicine if they had no participation in peer-reviewed or federally funded research? If the value to the university was evaluated only by patents and licensing income, and really most important, no contribution to information broadly available to the public? Because within the commercial world, there is certainly restrictions on access to the data. So I think we need to find this balance. What are some of the negative impacts of commercialization on the culture of university in this translation? Well, as we mentioned, that choose to do research on problems where a commercial pathway is more likely. The opportunity cost for faculty. I do think we have to recognize this is a long process for translation. And when we've asked our researchers in this area, what would you prefer, to hand off this potential intervention to another party to move ahead, or do it yourself? And about 80% of them said they wanted to do it themselves. That is, it's a long task, and you could argue, is that the most efficient use of resources for the society, or should there be a better way to move it on to the next group that's really helpful in that translation? We worry about research practices, particularly data withholding, the issue of hyping results beyond the data, which I certainly feel the need to keep a watch on, because I think it's pretty obvious that when faculty members have a more commercial interest, that tends to be the pattern. It's okay, but there has to be another group that puts some controls on it. And then the last thing would be the commitment to commercialization may be a barrier to new collaborations. You mentioned this is a team sport. It's a team science for translation to work. And, I'm seeing some examples where I think this actually can impair the ability of new collaborations to occur, and we have to watch that. So I've just thought a little bit about how do we optimize this academic entrepreneurship. Do we have a code of ethics or responsible academic entrepreneurship for faculty? Are universities assessing the negative as well as the positive components? And should there be some monitoring approaches to look at this? Even if I put at the bottom trainees. Trainees are oftentimes put in some awkward positions where they're asked to work on the research of their mentors, which may be more commercial. Some trainees love it. They go and work for that company after they're done. So I think there's lots of positives. I just feel we need, as responsible members of the research community and universities, need to keep track of it. So let me just tell you one way that we've done this is we've created translational research communities at our university. And really the goals of that were to bring researchers together across departments to role model successful translation within a university. Many times there are a couple investigators in individual departments that are trying to do that. And one of the positives I would see in the drug development community at Hopkins is really brought together our oncology group, our neurology group, and our clinical pharmacology group, which frankly were working quite separately in terms of trying to work through this complex maze. Help researchers with good ideas decide what role they should have in advanced translation, and then develop symposium targeted pilot cores that community looks at and says, this is what we need to be successful at our job. And these are the 4 translational research communities at Hopkins that we've developed, and I think we've sort of seen examples of most of these, right? The drug and vaccine development pathway, devices and procedural intervention, which is a large part of most medical schools, at least in the United States. Biomarkers and diagnostic tests, where we see the Telomeres group and her colleague at Johns Hopkins, Carol Greider, who works with that, and thinking about taking disease groups and say, how can we look at that? And talking, I guess I go back to saying, in some ways, in academic medicine, there's more of a tendency to just develop a new biomarker rather than to really test the utility of that biomarker and get it to be a diagnostic test that is well recognized. And then finally, not forgetting behavioral healthcare delivery and social system interventions, where those researchers are constantly, feeling that they've developed a good intervention, it hasn't disseminated, and what are the tools that we need to give those researchers as well. So, in summary, universities have worked for centuries to develop a culture of discovery for fundamental truth that I think needs to be preserved in this era of translational medicine. There are definitely opportunities in academic entrepreneurship that really change the mindset and create a huge enthusiasm for our faculty that we need to help foster. But we do think that universities should develop some way to systematically monitor this process to make sure that the university and the commercial translation stays in a positive light and meets the needs of the public who have great expectations for these partnerships. Thank you.
Thank you so much for a wonderful talk. That concludes our session. We're slightly over time. I'd like you to join with me in thanking all our speakers for absolutely wonderful talk. And we look forward to hearing more of the progress of all their translations. So thank you.